Naveen, we shall discuss about the minor experiments.
Yes,
Okay.
ma'am.
As per your university, there are 4 minor experiments: hemoglobin estimation, blood grouping and typing, bleeding time, and
Clotting
clotting
time.
time.
Yes, ma'am.
Okay, fine. So we'll start with hemoglobin estimation,
Yes, ma'am.
right? Now, what is the name of this method that you are using in physiology lab?
Salis acid hematin method, ma'am.
Salis acid hematin
Salis.
method. Many students, they will not know to pronounce this name also. They used to say Sahelis.
Yes.
Okay, just read on the hemoglobin tube, that hemoglobinometer tube. In that it will be encrypted as Salis.
Oh,
Okay, if you don't uh, just in that second, if you don't remember that name, just read on that and you can
yes,
tell
ma'am.
the examiner, right? Okay, what is the principle of this method?
Mm. So the blood is converted into acid hematin
Hmm.
after adding n by 10 of HCl to it. So after adding the distilled water to the acid hematic mixture, it converts the color into brown color, ma'am. So the brown color indicates the concentration of the hemoglobin, ma'am.
Okay.
So these color is compared with the comparator given along the hemoglobinometer, ma'am.
Yes.
And once we get the exact matching, the markings in the hemoglobinometer tube is red in gram percentage, ma'am, to
Okay.
estimate the hemoglobin.
Okay. So for mixing, after you add the distilled water drop by drop, you will have to mix the
Yes, ma'am.
contents, right? There will be a stirrer provided for the mixing.
Yes, ma'am.
Will you read the values or the graduations with the stirrer inside, or if you will take it out?
No, we'll take it out, ma'am.
You will take it out. Very good. Okay, fine. Can you add tap water for dilution purpose?
No, ma'am. It may create turbidity as it contains more salt, ma'am.
Mm.
So which might hinder the color matching, ma'am.
Okay. Can you add N by 10 HCl for dilution purpose?
Yes, ma'am. But
Okay.
it's not cost effective.
Okay. Many students used to say that if you add N by 10 HCl, the cells will get hemolyzed. But already the hemolysis have been done. All
Yes,
the complete RBCs have been broken down, and that's how we got acid hematin,
yes,
right?
ma'am.
So there is no problem in adding N by 10 HCl except that it is costly.
Yes, ma'am.
Right. Okay, fine. Now, one out of the way question,
Yes, ma'am.
Naveen. Why do you want to add acid to the blood, convert it into a brown color, and then compare with the comparator? Why don't you just compare the red color of the blood with another red comparator? Maybe a star question.
Mm, acid may lyse the RBC and the
You need
hemoglobin.
not lyse RBC.
I have no idea.
Okay, this is all related to your um, sensitivity of your color cones actually. Okay, your retina is more sensitive for the brown color
Oh,
rather than the red color,
okay.
that wavelength. Okay, so that matters.
Okay.
That's why we are converting that
Red
color into brown
yes,
by
ma'am.
adding the N by 10 HCl, and
Yes,
we are
ma'am.
doing that. Okay, what is a normal hemoglobin content in the blood?
So for the adult male, it is 14 to 18 gram per deciliter.
Mm.
For the adult female, it is 12 to 16 gram per deciliter, ma'am.
Okay, why it is more in males?
More RBCs, ma'am.
Okay, in children?
It will be more greater, ma'am.
Yes, it will be greater because more RBCs, again
Yes,
the same. Okay, is this Salis acid hematin method the accurate method?
I don't think so, ma'am.
Why?
As it is subjected to observer variation, ma'am.
Yes, to me it may be matching, to you it may
Yes,
not be
ma'am.
matching, so it is subjected to observer variability, right? So which is the accurate method then?
Gasometric method
Mhm.
and sign meth hemoglobin method using Drapkin reagent, ma'am.
Yes, good. So gasometric method, which is done using Wants-like
Once like
apparatus,
yes,
is
ma'am.
quite time consuming.
Okay.
But the ideal method is that, but routinely done accurate method is cyan meth hemoglobin
Okay,
method. Okay. Okay, fine. This ends the hemoglobin estimation.
yes, ma'am.
Shall we go on to blood grouping and typing?
Yes, ma'am.
Okay. What is the principle of this blood grouping and typing?
So basically, the blood grouping is determined based on the antigen antibody reaction, ma'am.
Okay.
So Once the antigen, that is agglutinogen, is present in the RBC, reacts with the agglutinin, that is the antibodies. It forms the agglutination reactions
Okay. Where is the agglutinogen present in the RBC?
in the surface antigen, ma'am.
Surface antigen. Okay. Where in the blood it is agglutinin is present?
The plasma.
Plasma, dissolved in the plasma.
Yes, ma'am.
Okay.
For the RH typing, it's based on the agglutination reaction, where the D antigen reacts with the anti-D antibody, ma'am.
Okay. So that's the principle.
Yes, ma'am.
Okay. What is Lanssteiner's law? Commonly asked question by the
Yes, ma'am.
examiner is this one. Lanssteiner's law.
So, if an agglutinogen is present in the RBC of the blood, the corresponding agglutinin must be absent from the plasma.
Okay.
So, if the agglutinogen is absent from the RBC of the blood, the corresponding agglutinin must be present from the plasma, ma'am.
Okay. Is there any exception to this law?
Yes, ma'am. RH negative people, they lack both anti-D antibody as well as D antigen, ma'am.
Yes. Excellent. Okay. Fine. Always remember agglutinogen and antigen are the same.
Same. Yes,
Gen,
ma'am.
gen. It ends with gen, both. Okay.
Yes,
Students will get confused with agglutinogen is antigen or antibody.
yes, ma'am.
Okay. So it is agglutinogen is antigen.
Okay.
The other one is antibody. Right. Okay. Can you tell me the procedure what we are doing in the lab to find out the blood grouping and typing?
Yes, ma'am. So the blood is mixed with the saline and forms a suspension. So then we'll take a tile and we'll separate into three. So in the first column we'll add anti-sera A, in second will be anti-sera B, and third will be anti-sera D.
Okay.
So we'll add few drops to each one of it, and we'll wait for 10 minutes, ma'am. So after 10 minutes, if we see the any agglutination is present, it's represent the positive for that particular reaction, ma'am.
Can you still more explain how you read it?
If in the first column it is precipitated,
Agglutinated.
then it is A blood group, ma'am.
Okay.
So if it is in B, it is B blood group. If it is in both AB blood group, if
Okay.
it is no, it is O blood group, ma'am.
Okay.
So in the D antigen, if it is present, it is RH positive. If it is negative, no, it is RH negative, ma'am.
Okay. Do you add any control group for this?
Yes, ma'am. We'll add the control group to assess any autoagglutination occurs by any external factors, ma'am.
Okay. So you'll just add only the saline
Saline.
suspension, leave it for 10 minutes.
Yes, ma'am.
Okay, fine. What are the other blood groups you know of?
Bombay blood group, ma'am, which has the H antigen, and MN system, Lutheran system, Lutheran system, and Defi
Okay.
and Kell.
Okay, fine.
Yes, ma'am.
Good. Many students will answer for this question, what are the other blood groups? They used to say A group, B group, AB group, RH group, like that. See, all this A, B, O and RH, they belong to one system called ABO system. Okay. Other systems means all this Bombay blood group based on H antigen, MN system, Lutheran,
ma'am.
whatever you have mentioned. Okay. You just remember the names. Okay, fine. What are the advantages of knowing the blood group?
So it's used for the identification of the person, ma'am.
Yes, every ID card has got the blood group
Yes, ma'am.
details.
And to avoid mismatch blood transfusion, and to know the risk factor of certain disease, like the A blood group is more prone to get cancer, and O blood group is more prone to get ulcers.
These are associated factors,
yes.
but actually not that causative association is not proved actually.
ma'am.
They are found to be more. Okay.
And also it's used in the analysis of forensic investigation and paternal disputes, ma'am.
Okay. What is cross matching?
Cross matching, it is a technique, it's done before every blood transfusion or any organ transfusion, ma'am. It is of two, that is the major cross matching and minor cross matching. In the major cross matching, the donor's RBC is reacted with the recipient plasma, whereas in the minor cross matching, the donor's plasma is reacted with the recipient RBCs, ma'am.
Which is very important?
Major, ma'am.
Okay. Okay, one last question in this blood grouping.
Yes, ma'am.
So you have identified the blood grouping and typing, you have made the tile and you have done the procedure.
Yes, ma'am.
Even after leaving for 10 minutes, you are still doubtful whether the agglutination is present or not.
Yes, ma'am.
How will you confirm it?
We'll take a drop of blood, and along with that we'll take antisera, and we'll put under the cover slip, and we'll visualize under the microscope, ma'am, to check
Okay.
when any agglutination is found or not.
Perfect. Good.
Yes, ma'am.